Hello. This one is starter 4, and by your recommendation and by the question of some students, I will try to prepare medical biochemistry tutorial video today. And this medical biochemistry can be prepared for health students, especially for pharmacy students, for nursing students, for medical laboratory students, as well as for medicine students. Therefore, these courses have actually medical biochemistry have three major units, and the first one, or the first unit, is all about protein metabolism, and the second chapter, which is very bulky chapters, is all about vitamins. As you know, vitamin can be classified into water soluble vitamins and lipid soluble vitamins. So today we directly consider only on the water soluble vitamins as part 1, and we can proceed to the next to lipid soluble, as well as all the components or the major subtopics of these chapters. So based on your recommendation and questions, I will try to depict or prepare this tutorial video for you. So you have to listen and use from this tutorial video. I'm sure that this tutorial video can be support you guys, so you have to listen attentively, and you have to share and subscribe for your fellow friends, so as to be benefited from the courses. So when you come to the introduction parts, as you know, vitamin is any group of organic substance essential in small quantities to normal metabolism, and this vitamin is found or required in micro level in our body, and they are very important at this minute amounts, so as to prevent us from different diseases. So vitamins are organic molecules which have a wide range of roles in the body, and the main function is as a coenzyme for enzymatic reaction. You know that coenzyme is an organic substance that used for biological catalyst with a couple of enzymes. So vitamin cannot generally be made by a human body, so must be provided in diet. So our body cannot be synthesized or produced this vitamin, so rather than we get this vitamin from the diet we are consuming in day-to-day for our life. So biosynthetic pathway for vitamins can be quite complex, so it is more energy efficient to take them from the diet. And to be called a vitamin, a substance must be fulfill at least these criteria. The first one is it must be organic. It must carry out at least one biochemical or physiological reaction in our body, and must be required in small amount or in minute amount in our body. Therefore, to be a vitamin, at least this criteria must be fulfilled. So when you come to cofactors, enzyme catalyze many reactions, but only in association with small molecule called cofactor, which could be described as a chemical tease, which can be what holds the receptor at the active site, that it means. So cofactors may be a metal ion such as zinc 2+ required for catalytic activity of a carbonic anhydrase. So cofactor can be organic molecule known as coenzymes, and this cofactor can also be a prosthetic group. Prosthetic group means that the group other than the active site, that means which are permanently associated with the protein, often by covalent bonds. Example, the heme group on hemoglobin, which can be used for carry oxygen through our blood cells, especially red blood cells. This heme is what a porphyrin having an iron molecule inside the porphyrin, and which is iron 2+, therefore it can be act as a cofactors. So a catalytically active enzyme cofactor complex is called holoenzyme, and the enzymatically inactive protein without the cofactor is called apoenzyme. Therefore, you have to differentiate holoenzyme and apoenzyme like this. So catalytically active enzyme cofactor complex is called holoenzyme, whereas the enzymatically inactive protein without the cofactor is called an apoenzyme. So these coenzyme coenzyme are irreversibly changed during catalysis. They are either modified or regenerated after the reaction. That means, so each kind of coenzyme have a particular chemical function. Some are oxidation reduction or redox agents. Some facilitate a group transfer. Example, the enzyme that is transferase enzyme can be used for facilitating what transferring of a group. So if we consider electron as a group to transfer, we can categorize all coenzyme as a being involved in transfer process. The number of important coenzyme is limited, but each of them may be associated with many different enzymes. Therefore, this coenzyme is also what used as a facilitator of chemical reaction as a catalyst. So as I mentioned before, enzyme can be generally classified into two: water-soluble vitamins and lipid-soluble vitamin. Water-soluble vitamin is mainly vitamin B in the C, but B may be B1, B2, B3, B5, B7, B6, B12, and then the B9 also, as well as vitamin C can be also a water-soluble vitamin, whereas soluble vitamin include ADK, vitamin A, vitamin D, vitamin E, and vitamin K. These all enzymes have its own roles in our body and have different sources of this vitamin from the food we consume. So water soluble vitamin is mainly vitamin B and vitamin C, and they are transported throughout the water medium of the body. This means they have a polar nature, not stored in our body, except for vitamin B12, which is cobalamin, and vitamin B6. So B vitamins function as a coenzyme. They must be modified before they can function. So vitamin C does not form a coenzyme and the function as it is. This means it does not need any coenzyme among the vitamin, which is water soluble vitamin, which does not need any coenzyme, is known as vitamin C. So many water soluble vitamins participate in energy metabolism also. This is another features of the water soluble vitamins. So these vitamins and have its own specific coenzymes. For example, vitamin B1 or thiamine have thiamine pyrophosphate as a coenzyme, and riboflavin or B12 have FAD and FMN as a coenzyme, and biotin have carboxybiotin, and the vitamin B12 or cobalamin have cobamide enzymes as a coenzymes. So each enzyme have its own coenzyme, but vitamin C does not have any coenzyme because it can perform its function as it is. So when it comes to vitamin B1 or thiamine, the structure seems like this, which is a C6 having a 2 nitrogen moiety, and the methyl group or CH2 group can be bridged the two pyrimidine and the thiazole coupled to each other. So the structure seems like this, and this thiamine is also known as vitamin B1 and has a central role in generating energy for carbohydrates. Therefore, this vitamin B1 or thiamine is what's very important for carbohydrate metabolism. So it is derived from the substituted pyridine and the thiazole, which is coupled by methylene group bridge, and the methylene bridge easily broken by prolonged exposure to heat and also cooking in alkaline solution like bicarbonate soda. Therefore, this vitamin B1 can be easily broke down by the prolonged exposure of heat, and also if you were cooking this vitamin B1 in alkaline solution or basic solution like bicarbonate soda, this methylene bridge can be easily disintegrate by this exposure that it means. So when you see the absorption, transport, and excretion of this thiamine is absorbed in small intestine by the carrier mediator system that is transported in the blood as thiamine, or in its coenzyme formed by red blood cells. So the storage of this vitamin is, as we mentioned before, is very poor. So excess is rapidly excreted in the form of urine from our body. This thiamine pyrophosphate is the coenzyme for vitamin B1, and this thiamine is converted to its active form, and the thiamine pyrophosphate in the brain and the liver by specific enzyme thiamine diphosphotransferase enzyme. So this enzyme can be used for converting the active form of thiamine pyrophosphate to in the brain and the liver by specific enzyme. That it means. So thiamine pyrophosphate is necessary as a coenzyme for the pyruvate dehydrogenase enzyme and the alpha ketoglutarate dehydrogenase catalyzed reaction, as well as translocatase transketolase catalyzed reaction of the pentose phosphate pathway. This one is also the carbohydrate metabolism pathway. So in that pathway also this coenzyme is very important. As we mentioned before, this vitamin B1 can be mainly important in carbohydrate metabolism. Therefore, this thiamine pyrophosphate also have its role in the carbohydrate metabolism as a coenzyme for vitamin B1. So this thiamine pyrophosphate as a coenzyme, so pyruvate to acetyl-CoA for citric acid cycle or Krebs cycle. So glucose can be converted to pyruvate can be converted to acetyl-CoA. This acetyl-CoA is what an intermediate for citric acid cycle or Krebs cycles. During that time, alpha ketoglutarate to succinyl CoA can be converted. So this alpha ketoglutarate can be converted to succinyl CoA, and a decarboxylation reaction, or decarboxylation means the removal of carboxyl group, which is released as a form of carbon dioxide of an alpha keto acids or a metabolite of amino acid. That means, so this transketolase is the thiamine pyrophosphate dependent enzyme which catalyzes the formation of 5-sugar component of RNA and DNA in the pentose phosphate pathway. So this thiamine is mainly found in a diet or a food we consume in day to day, like pork products and the sunflower seeds, watermelon, and the green beans, as well as asparges are the source of this vitamin B1 or thiamine. The thiamine is found in low amount in white bread, pork, cereals, and orange juice. So the dietary requirement for vitamin B1 or thiamine is proportional to the caloric intake of the diet and ranges from 1 to 1.5 milligram per day for normal adults. This one is the RDA of vitamin B1 or thiamine in healthy adults. That means, so if the carbohydrate content of the diet is excessive, then more thiamine intake will be required for the our body. That means, so sometimes the deficiency of this vitamin also can be risky for our health. So the deficiency in thiamine intake lead to several reduced capacity of cell to generate energy as a result of its role in this reaction, and the nervous system most affected as a tissue, as it uses a lot of chemical energy, and also the skin as well as gastrointestinal tract or GIT are badly affected as these regenerate frequently, requiring energy, because we say that the carbohydrate metabolism can be mediated by this thiamine pyrophosphate coenzyme. And the other clinical symptom of prolonged thiamine deficiency are related to cardiovascular as well as musculature defects, and the common disease associated with this vitamin B1 deficiency is known as beriberi, as a result of the diet that is carbohydrate rich and thiamine deficient. So an additional thiamine deficiency related disease is known as Wernicke-Korsakoff syndrome. This is also another disease associated with the deficiency of thiamine. So this beriberi or vitamin B1 deficiency, especially the clinical sign of such disease, is known as, for example, weight loss, apathy, loss of short-term memory, confusion, gastrointestinal tract problem, that GIT problem, that means irritability, peripheral neuropathy, as well as muscle weakness is a common sign of this disease. And the peripheral neuropathy means the impaired sensory motor, and the reflexes affect the arms as well as leg. So this beriberi can be classified actually into two: wheat beriberi and the dry beriberi. Wheat beriberi is oedema, also occurs along with enlarged heart and congestive heart failure, whereas dry beriberi is extreme muscle wasting, some symptom observed even after only 7 days of the thiamine deficiency. So this beriberi, especially wheat and the dry beriberi, can be what's associated with the deficiency of vitamin B1 or thiamine. So Wernicke-Korsakoff syndrome is linked to excessive alcohol consumption, and these alcohols have thiamine related problems. For example, poor diet to little vitamin consumed. Alcohol especially reduce the absorption of this thiamine, and increase the excretion of thiamine. Therefore, the ratio of absorption and the ratio of absorption and excretion is unbalanced. Therefore, due to that reason, such type of syndrome can be associated. So the most symptom of this disease can be what? Ataxia, or inability to control muscle movement, staggering gaits, and transient mental function, double vision across eyes, rapid eye movement, as well as memory loss and disorientation is the major symptom associated with Wernicke-Korsakoff syndromes. So the other one is riboflavin. Riboflavin have the structure of C6C3C6, having what? Nitrogen moiety, and this riboflavin has a three-linked 6-membered ring with a sugar alcohol attached to the middle ring, and the yellow color, or flavin, yellow in Latin. The name flavin can become from yellow, which means yellow means flavin, and it is the component of two coenzymes, flavin mononucleotide FMN and flavin adenine dinucleotide. These are the two common coenzyme for vitamin riboflavin, so involved especially in energy production. So this is the structure of what the structure of riboflavin. So when come to the absorption, transport, and excretion of this riboflavin, actually in our stomach there is a HCl acid release riboflavin from its bound forms, and absorption by the active or facilitated transport in small intestine can be takes place, and in the blood this riboflavin can be transported by a protein carriers. We know that we have different carrier proteins, so that carrier protein can be used for transporting these vitamins. Let's convert to coenzyme forms flavin mononucleotide and flavin adenine dinucleotide in most tissue, but mainly in small intestine, liver, heart, and the kidney can be what the place where this conversion can be takes place. So small amount of riboflavin is stored in our liver, and the excess riboflavin can be what removed from our body in the form of urine. So flavoprotein, the enzymes that require this coenzyme as a cofactor are called known as flavoprotein. So flavoprotein are an enzymes that require the two riboflavin coenzyme, flavin mononucleotide and flavin adenine dinucleotide. So several flavoprotein also contains a metal ion and are termed as metalloflavoprotein. Both classes of enzyme are involved in the wide range of redox reaction, like succinate dehydrogenase and xanthine oxidase are the common example of this metalloflavoprotein. During the course of the enzymatic reaction involving the flavoproteins, the reduced form of FMN and FAD are formed to be what FMNH to FADH respectively. This one is the enzymatic reaction of flavoprotein to produce this coenzyme. So from this FAD, the reactive site is the nitrogen, because of there is a lone pair on nitrogen, but the carbon having the oxygen or carbonyl carbon is highly electrophile because of electronegativity of oxygen is greater than that of nitrogen. But in case of nitrogen there is a lone pair, therefore this it become what reactive sites. So the two nitrogen can be act as what the most reactive site, no, for reduced form of hydrogen as on this nitrogen, that means. So adenosine monophosphate units can be attached as on the oxygen on the butyl group can be what attaches adenosine monophosphate units. So the main function of flavoprotein is what in the redox reaction it can be used for what taking electron from substrate and giving electron to substrate. Riboflavin cofactor is either reduced or gain of electron and the substrate is oxidized, whereas oxidized means loss of electron and the substrate is reduced. So riboflavin does this by moving hydrogen atoms, and the succinate dehydrogenase has riboflavin cofactors. Therefore succinate can be converted to fumarates. These two are an intermediate actually, and this takes place by the presence of what this fat or succinate dehydrogenase coenzyme, the cofactors. So the FAD has an electron acceptors. It can be act as electron acceptors. Yeah, because this alkane can be converted to alkene by giving off 2 hydrogen. Therefore, FAD acts as what? The electron acceptor here. So, and FAD containing an enzyme participates in breakdown of fatty acid to acetyl-CoA. The reaction is known as beta oxidation. So you know that this one from biochemistry 1 concepts. So FMN containing an enzyme shuttles hydrogen atom into the electron carrier chain, and FADH containing an enzyme glutathione reductase converts the oxidized form of glutathione to the reduced form. This glutathione is the best antioxidant enzyme that can be used for what? Abstracting hydrogen or snatching hydrogen, that it means. So this riboflavin also have found in diet. Actually 1/4 of the dietary riboflavin comes from milk product. Therefore the major source of this riboflavin is milk, and the rest from white breads, eggs, and the meat. And the food rich in riboflavin include liver, mushrooms, spinach, and other leafy green vegetables. Asparagus, milk, and the cottage is also at the source of this riboflavin, but milk is by far the dominant source of this riboflavin. So exposure to light causes riboflavin to break down. So milk and the cereals packaged in cartons to avoid what the exposure of light, so as to avoid what the breakdown of this riboflavin. Therefore RDA recommended daily amount is almost about 1.1 to that of 1.3 milligram per day for an healthy adults. Therefore this amount can be what decreased based on the sex, the age, and the health of an individual, that it means. Yes. So this riboflavin also have a disease, especially riboflavinosis is the disease associated with this riboflavin, and they are rare in the Western world due to the presence of adequate amount of vitamin in eggs, milk, meat, and cereals actually. So riboflavin deficiency is often seen in chronic alcoholic due to their poor dietetic habits. So symptoms associated with riboflavin deficiency include glossitis or inflammation of the tongue, that mean saporia or sebaceous gland disease, and angular stomatitis or inflammation of the mouth, and cheilosis or cracking of the tissue at the corner of the mouth, and the nervous system disorder, photophobia, as well as confusion is what the main symptom associated with riboflavinosis. Riboflavinosis is what the disease associated with riboflavin deficiency, and this riboflavin deficiency can be lead to what this disease with this phenomena or symptom. And this riboflavin function along with other vitamin B, so the deficiency of riboflavin can affect other metabolic pathways associated with this vitamin. So riboflavin decomposes when exposed to visible light, which can lead to riboflavin deficiencies in the newborn treated for hyperbilirubinemia by phototherapy. Therefore this one is a treatment for the deficiency of what? Riboflavin, especially newborn babies. The third vitamin is vitamin B3 or niacin. Niacin exists in two forms actually, nicotinic acid and nicotinamide based on the functional group attached on the benzene ring having a nitrogen, and both are found equally in food, and both forms performs what the function associated with niacin. Those active form are coenzymes nicotinamide adenine dinucleotide NAD+, and the nicotinamide adenine dinucleotide phosphate NADP. These are the active coenzyme for these vitamins. So both coenzyme NAD and NADP function as a cofactor for numerous dehydrogenase, example lactate and the maltase dehydrogenase, and the role is in redox reduction, oxidation reaction to produce what? An energy. So the absorption of this vitamin and the excretion, transportation of this vitamin can be also takes place in our body. For example, nicotinic acid and nicotinamide, the two common form of niacin, are readily absorbed from the stomach and the intestine by active transport and the passive diffusion. So almost all niacin is absorbed actually. So this niacin is transported from the liver to all tissue where it is converted to its coenzyme forms. The coenzyme form can be NAD plus and NAD P. So niacin coenzyme are stored in the liver and the excess is excreted as a variety of metabolite product in the form of urine. So it seems this NAD and the NAD plus can be seems like this, and the reactive site or the nucleophile is the one that having what carbon double bonds, and it can be what a prominent carriers of high energy electron like this. So this one is structures. So NAD sometimes act as electron acceptor, and simply the hydrogen on oxygen, the red hydrogen on oxygen can be what abstracted or accepted by NAD, so as to give what carbonyl carbon double bond plus NADH. So NAD and the NAD participate over this cellular reaction, especially those used to produce what an ATP. So this one is a reaction. The main function of NAD and the NAD is involved as an electron and hydrogen ion acceptor in glycolysis. Glycolysis means glucose to pyruvate. The conversion of glucose to pyruvate is known as glycolysis. This one is also the concept of biochemistry 1 actually. So that phenomena can be takes place supported by this coenzyme NAD. So under anaerobic condition mean the condition of without oxygen. So anaerobic condition, the resulting reduced form NAD H and the hydrogen is used in converting pyruvate to lactate, and at the end of the day it can be generating what NAD plus. So this pyruvate lactate can be an intermediate actually, and this intermediate can be what interconverted by using these coenzyme as well as cofactors. So NAD also reduced in many reaction in citric acid cycle or Krebs cycle. It also reduced in the reaction with which converts alcohol to acetaldehyde. So NAD pH is reduced form of NAD P plus, and used in the pathway for the fatty acid synthesis. Cell which synthesize a lot of fatty acid, example in the liver, have a higher NAD pH concentration than those which do not have meaning, like muscle cells for example, have what a less amount of what this NAD pH that it means. So reductive biosynthesis of fatty acid, as you know, this fatty acid can be biosynthesized from acetyl-CoA and the malonyl-CoA, and acetyl-CoA is as starter unit which have 2 carbon unit that mean, and the malonyl carbon which is a source of 3 carbon, and one scenario of fatty acid synthesis is that one, but there is also reductive biosynthesis technique that's supported by NAD pH as electron as is the electron and the proton donor. It can act as electron and the proton donor, this NAD pH, which is have very important role in biosynthesis of fatty acids. So this niacin is not true vitamin, as it can be derived from an amino acid tryptophan, and this niacin can be act as what a false vitamins. However, the ability to utilize tryptophan for niacin synthesis is inefficient, which means that 60 mg of tryptophan are required to synthesize 1 mg of niacin. Also, synthesis of niacin from tryptophan requires another vitamins, vitamin B1, vitamin B2, and vitamin B6, which could also be limited on the marginal diet. Therefore, the biosynthesis of niacin can be what takes place by tryptophan, even if it is inefficient. But only tryptophan cannot be what produce niacin, rather than you have to use what vitamin B1, vitamin B2, and vitamin B6 for synthesis of niacin, or vitamin B3. So the recommended daily requirements or RDA for niacin is 30 to 90 mg per day, and this is expressed as niacin equivalent to include niacin formed from of hands. So rich source of niacin are mushrooms, wheat, prawns, tuna, chicken, turkey, asparagus, and peanut. These are the major source of what vitamin B3. So animal protein except gelatin are rich in tryptophan, as are egg and milk also the source of this vitamin B3. So diet with corn as staple food, corn containing niacin but not much as absorbed as it is bound to the protein. Soaking the corn in alkaline solution, this means lime water for example, release the bound niacin and it can be what absorbed. Therefore niacin is a heat stable, so little is lost in cooking. Therefore this vitamin, what makes this vitamin odd is it is actually thermostable, so heat cannot be easily break the niacin. So this niacin deficiency can be always associated with pellagra, the disease known as pellagra, which have what the symptoms of depression, dermatitis, and diarrhea. This disease can be associated with this niacin deficiency. So the clinical evidence of pellagra develops about 50 to 60 days after the start of the niacin deficiency diets. So the diet deficient in niacin means that the tryptophan, or the amount of tryptophan or vitamin and amino acid can be what decreased. So this lead to what inflammation of the tongue, dermatitis, weight loss, diarrhea, depression, and dementia also what associated with the deficiency of this niacin vitamin. So this picture depicts how a pellagra can affect your dermatitis or your skin like this. So this one is a deficiency of niacin or vitamin B3. Niacin and the nicotinic acid deficiency in several physiological conditions like heart nerve disease and the malignant carcinoid tumor, as well as certain drug therapies, example isoniazid and antituberculosis, can lead to the deficiency of syndrome. Yes, adverse effect of such drug can also be what lead to this niacin and the nicotinic acid deficiency. So here heart nerve disease is a genetic disorder and the symptom is similar to pellagra, and here tryptophan absorption is actually impaired, tryptophan conversion to niacin also impaired, and the malignant carcinoid tumors, tryptophan metabolism is altered, resulting in excess serotonin synthesis in our body, and this one leads to the disease associated with niacin and nicotinic acid deficiency like heart nerve disease. Actually this heart nerve disease can be is what a genetically inherited disease, and malignant carcinoid tumors also associated with this vitamin deficiency. The main pharmacological use of this niacin or vitamin B3, niacin acid, especially nicotinic acid, rather than nicotinamide, in pharmacological doses of about 2 grams per day lower the plasma cholesterol level and has been shown to be useful therapeutic for hypercholesterolemia. Therefore it have the ability to reduce the cholesterol in levels in our body, especially the nicotinic acid rather than nicotinamide. So the major action of nicotinic acid here is a reduction in fatty acid mobilization from adipose tissue. Although nicotinic acid therapy lower blood cholesterol, it also causes what the depletion of glycogen store and the fat reserves in skeletal as well as cardiac muscles. So this vitamin is very important in the form of nicotinic acid. So also there is an elevation in blood glucose and the uric acid production. So nicotinic acid therapy is recommended for diabetics or sufferers of guts. So toxicity effect, especially flushing of skin, itching, nausea, liver damage can also the toxicity effect of this vitamin B3 or niacin. So the next one is vitamin B5, which is known as pantothenic acid. The structure seems this one. So it is an amide of beta-alanine and the pantothenic acid actually. And the pantothenate is required for synthesis of coenzyme A, and this is coenzyme A, and it is the component of an acyl carrier protein ACP, domain of fatty acid synthesis. So pantothenate is therefore required for the metabolism of carbohydrate via a TCA cycle or triacetic acid cycle or Kookeef cycle, and all fats and the proteins can be here important for this cycle. So at least 17 enzymes have been identified as required coenzyme or ACP, acyl carrier protein derivative for their function. So this is the vitamin B5 or pantothenic acid. Phenomena. So the structure seems this one. It have what the beta markup to ethylamine unit as moiety attached on it, and there is a pantotenic unit. So pantotenic acid is converted to coenzyme A by combining with the parts of amino acid cysteine and the derivative of adenosine diphosphate or ADP. This coenzyme A and acetyl-CoA is an essential for formation of ATP from the breakdown of carbohydrate, protein, alcohol, and fat. So forms acetyl-CoA from acetate, which is a metabolite of this process. This allows acetyl-CoA to enter the citric acid cycle, generating ATP. Therefore, ATP can be produced from these intermediates, acetyl-CoA. Acetyl-CoA can be used for fatty acid synthesis as well as for amino acid synthesis, and it can be obtained from either alcohol or a glucose metabolism. So acetyl-CoA can also be condensed with carbon dioxide to begin the synthesis of fatty acid. As I mentioned before, acetyl-CoA is always the starter unit for the synthesis of what? Fatty acid. So acyl carrier protein, ACP, acyl carrier protein attached to the fatty acid and shuttles them through the metabolic pathway, which increases their chain length, which increase what? The chain length. Therefore, acetyl-CoA is, we say that acetyl-CoA is the starter unit, whereas malonyl-CoA is what? The extender unit. But acetyl-CoA is a source of two carbon in the bracket, as you see, and the malonyl-CoA is a source of 3 carbon. But the first fatty acid that can be synthesized by this mechanism have what? 4 carbon. So where is 1 carbon goes during this process, it can be what? Lost in the form of carbon dioxide or decarboxylation. This pantotenic acid is commonly found in meat, milk, and many vegetables, and mushrooms, liver, peanut, eggs, and yeasts, broccoli, and the milk also at the major source of this vitamin, and the adequate intake for adult is maximum 5 milligram per a day. This vitamin is very important again. So this deficiency can also lead rare effects due to its widespread distribution actually in green cereals, legumes, and the meat. So the deficiency of pantotenic acid is extremely rare. So deficiency may occur in alcoholism, where very nutrient deficient diet occurs. So the symptoms of pantotenic deficiency are difficult to assess since they are subtle and resemble those of the other vitamin B deficiency. So it is vitamin B5 deficiencies. Actually, it is difficult to identify because it seems what the deficiency of another vitamin B also. So experimental trial in humans showed headaches, fatigue, impaired muscle coordination, GIT tract problems, and they are among the few symptoms of the vitamin B5 or pantotenic acid deficiency. So when it comes to this biotin is found in two forms: free vitamin and the protein-bound coenzyme form biocytin. So a bond between the carboxyl group of biotin and lysine enzyme, amino acid in the protein is made to form biocytin. So biotin is absorbed from small intestine, biocytin is not. Biocytin can be converted back to biotin by the enzyme biotinidase, which cleaves the bond linking biotin to the protein. That is mean. This biotin action or vitamin B7 is the cofactor required for the enzymes that are involved in the carboxylation reaction. Example, acyl-CoA carboxylase and the pyruvate carboxylase can be the two cofactors required for the enzymes that are involved in carboxylation reaction. So carboxyl group of biotin covalently attached to the lysine residue in the enzyme, and the addition of carboxyl group makes a carboxybiotin enzyme intermediate where the enzyme where the new carboxyl group is bound to the N1 of the biotin ring. So this new carboxyl group is then transferred to the substrate. So structure of carboxybiotin seems this one. It have two head in the tail. Actually, it seems head in the tail. The head one is activated carbon dioxide, whereas the tail one is what? Lysine. And the carboxyl group of biotin bind to the amino acid lysine biotin, and this carboxyl group added to N1 of the carboxybiocytin, and this carboxyl group is also transferred to what? the substrate. So acetyl-CoA carboxylase catalyze carboxylation of acetyl-CoA to form malonyl-CoA in fatty acid synthesis, and the pyruvate decarboxylase catalyze addition of carbon dioxide to pyruvate to give oxaloacetate, an intermediate in citric acid cycle. So other biotin carboxylase break down the amino acid leucine, theronine, methionine, and isoleucine for energy needs. So biotin is required for metabolizing fatty acid, carbohydrate, and what? Amino acid. So mainly this biotin can be found in different foods like whole grains, eggs, nuts, and legumes, and the adequate amount of this vitamin required for daily consumption is almost 30 milligram per day. So it is also synthesized by intestinal bacteria, and as such deficiencies of the vitamin are rare because in our intestine the bacteria can also be what synthesize these vitamins. So deficiencies are generally seen only after long antibiotic therapies which deplete the intestinal fauna, or following excessive consumption of raw or eggs. So the latter is due to the affinity of the egg white protein avidin for biotin, preventing intestinal absorption of biotin. So avidin is denatured by cooking actually, and this means at high temperature this avidin can be what? Denatured because it is proteins. So eating raw egg and the biotin deficiency has been reported in alcoholics. Eating only 3 raw egg per day is very recommended. So the deficiency of this biotin or vitamin B7 associated with genetic defect like low amount of biotinase can't release biotin from biotin. So biotin deficiency may develop if a low biotinase level is detected. So symptoms include like skin rash, hair loss, convulsion, other neurological disorder, and impaired growth can be what the symptom of this biotin deficiency. So this it can be treatable. Actually, this biotinase deficiency can be treated by using simply what the vitamin itself. The vitamin itself can be used for what? Treating this deficiency. So this one is the question for you. So you have to try by yourself. For example, why is important? Why it is more nutritious to eat raw vegetable than boiled vegetables? Because mainly raw vegetables have vitamins, as you see. So these vitamins, some vitamin can be what thermolabile, so it can be maybe easily can be what break down, and the nutritious value can be what decrease. Therefore, by this principle, you have to what answer this exercise by yourself. And when you come to this vitamin B6, this vitamin B6 have different name like pyridoxine, pyridoxal, and the pyridoxamine based on simply the functional group attached to the rings. Pyridoxal means an aldehyde functional group, pyridoxamine means an amine functional group, and the pyridoxine having an OH functional group on H2. So are collectively known as vitamin B6, and all 3 compounds are phosphorylated to biologically active form of vitamin B6. Pyridoxal phosphate or PLP is what the coenzyme for vitamin B6. So this conversion is catalyzed by the ATP requiring enzyme pyridoxal kinase. So both the coenzyme form and the free form of vitamin B6 can be easily absorbed, and they are transported to the liver via the portal blood where phosphorylation can be takes place. So the coenzyme is then released from the liver into general circulation, bound to albumin or the blood protein for transport. Therefore, vitamin B6 is mostly stored in the muscle, and the high level X, if it is above optimum, it can be what in the same manner it is created in the form of urine like other vitamins. So this pyridoxal phosphate function as a cofactors in enzyme involved in transamination reaction required for the synthesis and the catabolism of an amino acid, glycogenolysis as a cofactor for glycogen phosphorylase, neurotransmitter synthesis is also another role of vitamin B6. Catalyzing step in heme synthesis, heme which is in hemoglobin porphyrin, that it means, and other vitamin formation can also done by this pyridoxal phosphate. So this vitamin B6 and amino acid metabolism, this PLP participate in the reaction in the form of non-essential amino acid, and this pyruvate acceptor can be converted to alanine in the presence of alanine aminotransferase, and this glutamate can be converted towards alpha ketoglutarate in the presence of glutamate aminotransferase. Therefore, PLP is also responsible for interconvention of D and L amino acid, which is act as a racemase. So it can also racemase means simply racemic mixture, meaning the amount of D and L must be what 50-50, that it means. So it can also be involved in the conversion of homocysteine to cysteine, which occurs during methionine metabolism in our body. So glycogen is a large polymer of glucose, and the PLP is a coenzyme for glycogen phosphorylase, which is required for glycogen breakdown. Therefore, glycosyl residue are sequentially removed and phosphorylated, and this helps keep the steady blood glucose level or blood concentration. Therefore, this vitamin B6 also have what a great role in DM or diabetes mellitus also. And this B6 can also be used as a neurotransmitter synthesis. Amino acid can also be used to produce non-protein nitrogen compound, and many of these are a neurotransmitter, which is very important for brain function, that it means. For example, serotonin from tryptophan, dopamine and norepinephrine from tyrosine, histamine from histidine, and gamma amino or butyric acid or GABA from glutamic acid. These all can be what a neurotransmitter, which is very important for our brain function. So B6 prescribed for the patient with psychological problem also, because it have what the healing effects for neuropsychological trauma or infection for disease can. Yes. And this to also be used for heme synthesis. So alanine cyanidase catalyze a step in synthesis of heme which require PLP. So this vitamin B6 and the vitamin formation also, PLP plays a key role in converting tryptophan to what niacin. So this PLP. So this PLP plays a role in converting what tryptophan to niacin. So this vitamin also mainly found in our diet, but the maximum amount for normal adult is 1.4 to that of 2 milligram per day. During pregnancy and lactation, the requirement for vitamin B6 increase approximately to 0.6 milligram per day for pregnant women, and the one that lactation, or the one that producing milk, that I mean, yeah. So this vitamin B6 is stored in animal muscle to found in meat, fish, and poultry. Poultry also as a source of this vitamin B6. Also found in whole grains, carrots, potato, spinach, and the banana, as well as avocado is the main source of what vitamin B6. Not stable under heat as well as alkaline condition. This vitamin also is highly heat sensitive, and it can be resist to at a basic condition or basic environment. Sometimes this vitamin may lead different disease if it become deficient. So the deficiency of this vitamin B6 leads to anemia, which is associated with heme deficiency, which means iron deficiency, that it means dermatitis relates to niacin deficiency, and the convulsion, depression, confusion, reduced immune response, which is PLP needed to form certain types of white blood cells, and the peripheral nerve damage can also be associated with this vitamin B6 deficiency. So deficiency of vitamin B6 are rare, and usually are related to what an overall deficiencies of all the B group vitamins. Some alcoholism also, alcohol metabolism produces acetaldehyde, which decrease PLP formation, and isoniazide or anti-tuberculosis medicine, and the penicillamine used to treat rheumatoid arthritis and cystinuria are the two drugs that complex with pyridoxal and PLP, resulting in deficiency in this vitamin. Therefore, some drug can also have an adverse effect on this vitamin B6 that it means. For example, the drug that given for anti-tuberculosis, isoniazid, and the penicillamine used to treat rheumatoid arthritis, cystinuria, are the drugs that can have an effect on this vitamin B6 deficiency. The main pharmacological use of this vitamin B6 is carpal tunnel syndrome or nerve disorder in wrist, sometimes treated with vitamin B6, and premenstrual syndrome could be also treated with this vitamin B6. And the toxic effects, for example, long-term intake can lead to irreversible nerve damage. Yeah, the toxic effect of this vitamin B6 means long-term intake can lead to irreversible nerve damage can also be associated. So the next one, the most important one, is folic acid. This folate is a generic term, include folic acid and the various forms of vitamin found in natural foods. This folic acid, also known as folate monoglutamate, is the form of absorbed in the body, and this is the form found in fortified food and the supplements. And most of the time, this folic acid can be given for pregnant women. So most folate found naturally in the food contains additional glutamate molecule attached to the carboxyl group, which actually decrease folate bioavailability. So yes, the bioavailability or the F value of the folate in our blood can be compromised. So these extra glutamate residue are linked to one another by the modified peptide bond involving the alpha amino group and the G carboxyl groups. Yeah, so the structure of folic acid seems like this. You have to see from this diagram. And the folic acid formation, folic acid is formed from three distinct parts. One is pyridine rings, 6-methyl pyridine, and the para-amino-benzoic acid PABA, and the glutamic acid also these three distinct parts that used for folic acid formation. The 6-methyl pyridine links to PABA to form butyric acid, and the glutamic acid residue conjugate to this acid to form a folic acid. This one is the mechanism of formation of folic acid in our body. So this seems the mechanism. So 6-methyl pyridine and PABA, para-amino-benzoic acid, can be react with glutamic acid to form this folic acid. And these folates polyglutamate forms from food must be hydrolyzed to the monoglutamate form in the gastrointestinal tract, and the folate conjugate enzyme removes the excess glutamate residue from our blood, and the monoglutamate form then transported actively across the intestinal wall. Especially removal of glutamate makes folate less negatively charged so passes through the membrane more easily, and the large doses of folic acid from supplement will passively diffuse through that's by diffusion that I mean. So this folic acid or folate can be easily cross the intestinal wall and the cell membrane as well. So monoglutamate form goes to liver via the portal blood draining in the small intestine. Here it is charged back to polyglutamate polyglutamate form, and this is stored in the liver or released into the blood or bile. The folic acid is mainly excreted in the urine as a metabolic product that can also be excreted into bile and reabsorbed by enterohepatic circulation. So the intestinal bacteria synthesize folate, but this may all be excreted in the feces, the forward. So circulation of substances such as bile salts, which are absorbed from the intestine and then carried to the liver, where they are secreted into the bile and again enter the intestine. That means this phenomena is known as interhepatic circulation. So interhepatic circulation means simply circulation of substance such as bile salt, which are absorbed from the intestine, they carried to the liver, where they are secreted into the bile and again enter to the intestine. This phenomena is simply known as interhepatic circulation. This phenomena is known